professor of Pediatric Hematology Oncology, as well as he have a lot of publications regarding targeted treatment with CAR T-cell in B-cell acute lymphoblastic leukemia, and that's the name of his talk. Okay.
Thank you very much for the kind introduction. So the talk of today will not be on the result of CAR T-cell in BALL, because I think everybody is aware of that. The idea is just to focus on what we can do to improve the results that are, of course, amazing, but they are not perfect until now. So I think Emily is one of the most famous kids in leukemia meeting. Was the first patient treated in Philadelphia with a nice success and a very long persistence of CAR T-cell. As an introduction, I think the result, you know, the complete remission is excellent, around 60 to 80%, depending on the trial. What I want to focus your attention is that we have to be careful to compare the different studies, because each study has different virus, different production in the GMP. So when we compare study of CAR T-cell, we have to really pay attention on all these details. So just a slide to remind you what is a CAR T-cell. So chimeric antigen receptor. Chimeric doesn't mean that we don't have this receptor in natural life. So it's a construct from the variable region of the antibody, and then you have a, I will try to use the pointer, and then you have a transmembrane domain, and then you need two important domains. One is an activation domain, most of the time is the CD3 zeta, and then you have a co-stimulatory domain who can be CD28 or 41BB to really improve the proliferation of the CAR T-cell. So when you have these two domains, here you have a second generation of CAR T-cells, so you have or 41BB or CD20, and if you have both, you have a third generation, and now we have also fourth generation and fifth generation of CAR T-cell, and you can introduce also some interleukin activator. So this is the process, or if you want, the schema of CAR T-cell administration in a patient. So first step, you need an apheresis, a T-cell apheresis from the patient, and looks simple, but you have to remember that most of the time our patient with multiple relapse LLL, or sometimes with 2 or 3 bone marrow transplantation before, so sometimes it's not so easy. But until now, all the study had proven that we can apherese these kids and have also quite good T-cells. Then the second step is the transduction of the T-cell with a vector that could be a retrovirus or a lentivirus, and then you have the expression of the CAR on the T-cells, and then what you need, a period of expansion of the T-cells, so a culture, and then very important, you need all the quality control before infuse your CAR T-cell to the patient. So these are the result of the more important study. I want just to focus you on the last part of the table, and if you can see the complete remission rates, it's amazing, but still 1 year after infusion you have almost 50% of the patient who relapse. So there is still improvement. So here you can see the Novartis trial. You can see that after 1 year, around 50% of the patient relapse. And on the right, the curves come from the Seattle experience that go until 4 or 5 years, and still they have also late relapse after CAR T-cell infusion. So what we can do to improve these curves and to try to avoid relapses. So first barrier is that Still, 20–30% of the patients are not able to receive the CAR T-cell because they are too sick to wait 4 or 8 weeks until the product is ready for infusion, and so there are still work on that. If you look to the Eliana trial, amazing result, complete remission around 80%, but if you look in details, the result of the trial, 17 patients were not able to receive the CAR T-cell. So if you calculate the intended to treat analysis, the overall remission is more around 66% of the patients. So it's an amazing trial, amazing result, but still there is this issue. So one solution could be academic CAR T-cell production. This is a Milteni machine. Germany is conducting a trial on that. I don't know if the solution, but we will see it's an automatic CAR T-cell production, so you just put the T-cells and then everything is automatic. Another solution could be off-the-shelf products. The first report was from Gosh in London. So they report 21 patients and 7 were kids. And as you can see, what they have done, that they knocked out the CD52, you will see after Y, and also the TRAC gene to avoid the HLA recognition of the T cell. So if they gave to the patient alemtuzumab to have really a complete T cell depletion, they have better result with a complete remission of 82% of the patient. For the reason they knocked out the CD52 on the CAR to avoid that alemtuzumab also destroy the CAR T cell. They had only one cutaneous GVHD, otherwise no GFG. So the problem is that the graft of the CAR T cell it's very short, so it's until now just a bridge to transplant. Another solution coming out is to use NK cells. So with NK cell you have not the problem of HLA recognition because they don't need it. They are independent from it to recognize the target. The problem here is also the persistence of T cells. Now there are many groups working on that and trying to get NK cell for cord bloods, and some trials are ongoing with this kind of product. Another issue is, of course, to try to avoid this 50% of relapse. There are many causes of relapse. The first one are very early CD19 positive relapse. So the cause is the lack of the persistence of the CAR T cell. We will see after what we can do on that. And the second type of relapse are CD19 negative relapse, and are most of the time late relapse after 6 months. And could be, the cause could be mutation, alternative splice, or downregulation of the expression of the antigen. And the third cause can be lineage switch. We saw that and was already published from many groups that MLL infant patient can switch to a myeloid leukemia after receive a very important pressure on the CD19. And most the event occur during the first year of the follow-up. So what we can do to enhance persistence of the CAR T cell to avoid relapse? First thing, costimulatory factor will be an important player. As you know, we can have for 1BB and CD28, the Eliana trial and the Kymriah is for 1BB co-stimulator CAR, and what we saw that there is a longer persistence compared to CD28. But if we take the example of the adult work with the diffuse large B-cell lymphoma, they use more CD28 CAR, like the CAR from Kite, and there you have probably a lower persistence but a stronger effect on the lymphoma with better results. But in leukemia, probably the 4-1BB until now is probably the best CAR, because you really want a longer persistence of the CAR. If you take the example of the NCI study with CD28, they were able to put the patient in remission, but if the patient was not transplanted afterwards, normally he relapses. Another component which is very important is lymphodepletion before the infusion of CAR T cell, and you can see here from these two trials in Seattle, patients who receive fludarabine and cyclophosphamide have a better survival compared to patients that got other lymphodepletion, most of the time only cyclophosphamide. So lymphodepletion is important. I think that everybody now is aware that fludarabine and cyclophosphamide are the best lymphodepletion for this kind of treatment. Also the starting T cell phenotype play a role, probably more specific lymphocyte like central memory T cells, stem cell like memory T cell, if are present in the product before going to manufacturing, they can improve the persistence of the CAR after the infusion. There is a nice paper from Frayet et al. It's an experience with adults with chronic lymphocytic leukemia, but still a very nice paper showing that the patient who had CAR T cell with more expression of CD27, CD8, but very low expression of PD-1 are predictive of a good response. Here more details of this study. If you can see, this is a transcriptome analysis. You can see that the patient who have a complete remission or partial remission have more early memory gene expression, have more memory here or C, and you can see they have less sign of exhaustion with less expression of gene of exhaustion. So this is very important, the starting T cell phenotype, and they were also able in this chart to show that expression of PD-1, TIM3, and LAG3, all markers of exhaustion. You can see that the patient who express a lot of this marker have a non-response to the CAR T treatment. Here also the example from Seattle, the same patient who express PD-1 are more frequently having a relapse. Here also, if you look to the persistence, so B-cell aplasia, you can see here in this chart that if the patient have a presence of the CD19 antigen in the bone marrow, that could be blast or also normal B cells, have a better persistence of CAR T cells. So doesn't mean that the antigen expression is also important for a persistence of CAR T cell. So in this direction, the Seattle group decided to try to do a trial with a vaccination, to try to expose the CAR T cell to the antigen and to have a better persistence. What they have done that with the rest of the T cell that were apheresis for the CAR T cell production, they transduce this cell with a CD19 antigen truncated, and then they have a T cell who express the CD19, and then what they have done, they presented last year at ASH, what they've done, they infuse the patient with the CAR here, and then you have an expansion in the blue line of the CAR T cell. You have a very good response with B-cell aplasia, and then every 4 weeks they infuse the CD19 T cell expressing cell, and then you can see here that you have expansion of the CAR T cell after the infusion of what they call TAPC, T cell antigen presenting cell. So this could be a solution for patients who have maybe only MRD at the moment of the infusion of the CAR T cell. So you give this CAR T-cell infusion to really maintain CAR T-cell, the CAR T-cell in the blood. Then another solution could be a fully humanized CAR T construct. So we know that probably using a murine CAR, you can have some CAR T cell rejection from the immune system of the patient. Here the experience of CHOP, you can see the infused patient that already received CAR T cell and they were having a CD19 positive relapse. You can see that they were able to put this patient a second time in remission. What was very interesting is that the patients were CAR naive. They had a complete remission of 100% in this patient and leukemia-free survival around 82%. So very, very interesting results. So now if we move to the second cause of relapses after CAR T-cell infusion. One thing that it's important, and maybe if you have CD19 negative relapse, try to have a multi-antigen target. So the first trial that was published was the experiment from NIH with a CAR against CD22. So they infuse 21 kids and 17 adults. And the complete remission rate was 73%, so quite interesting, but the median remission duration was only of 6 months, so there was clearly a problem, and was very well described in the paper that this patient, under a pressure of CD22, the CD22 site density really go down, so this leukemia cell express less the CD22, so afterward this patient relapsed, so there was a really short persistence of the remission. So what's next? So many groups in the US and also in Europe and China, they focus their future trial on dual CAR. You have many possibility to do that, so you can give a co-infusion of CAR, so you give a first infusion of CD19 CAR and a second one anti-CD22. You can have a co-transduction. Doesn't mean that you will have some cells expressing only CD19 CAR, some cells expressing CD22, and some cells expressing both CAR. Or the third solution could be to have a bivalent tandem CAR, so you will have the CARs one after the other. Here just a table summarize the trial, the most important trial that are ongoing and then were updated during ASH last year. You can see this is the Seattle group with a co-transduction CAR T-cell. Then you have the group from the NCI and Stanford with a bivalent tandem, and then a hospital with co-infusion in China, and the group from Gosh in London. You can see that the complete remission rate is very good, very comparable to the CD19 CAR. Now it's too early to know if this patient will relapse CD19 or CD22 positive. We have to wait probably one more year. But last year, at the end of the year, was published the first trial from China. In China, they have many trials on CAR T-cell. It was a co-infusion trial, and what was very interesting in this publication that the patient who relapsed, the 24 patients who relapsed, 23 over the 24, so almost all the patients were CD19 and CD22 double positive. Doesn't mean that there was really not a persistence of the CAR, so they were able to avoid the CD19 and CD22 negative relapse, but the persistence of the CAR was not good. So still work to do in this dual targeting strategy. And then what we can also do to improve the durability of the remission is to improve functionality of the CAR T-cell, and then it's really an amazing field. I will just give you some inputs. I'm not able to explain all the research that is done and is ongoing in this field. So you can associate the CAR T cell to checkpoint inhibitors. You can also knock down the PD-1 in the CAR, or you can also have modular switch design with an on-off CAR T cell. Just some example. First one, the experience of CHOP with the use of checkpoint inhibitors. What they were able to show that patients who had get a response and then they have an early B cell recovery, doesn't mean normal B cell in the blood, or they have a very important extramedullary disease, they really respond to checkpoint inhibitors. But patients who have not responded to CAR T cell at the beginning with a bone marrow who is still positive for blast, they are not able to respond after adding checkpoint inhibitors to the CAR. So for further investigation are needed, but I think it's a very interesting field also for lymphoma. And of course you have to pay attention of GVHD, because as you know, checkpoint inhibitors can really increase GVHD in patients who got already a bone marrow transplantation. Another strategy will be the switch on-off CAR T cell. So basically what they are able to do, that you will have a CAR with only an activation domain, and you will have a second domain with the CD3 zeta that is activator, so you will need always an activation and a co-stimulation. And when you give a small molecule as rapamycin, you have a heterogenation of the CAR, and then you have the activation of the CAR T cell. And you can see on the chart here that if you give more and more of rapamycin, you have a better response against the tumor. So it would be interesting to try to give some rest to the CAR T cell. It would be interesting to try to avoid some side effect if the cytokine storm is too important. So just a few slides on the role of bone marrow transplantation after CAR T cell therapy. So here the experience from the Seattle group that was presented at ASH. You can see on the left that patient seems to have better result with consolidation with hematopoietic stem cell transplantation, and who are the patients who really benefit from that are patients who has never received a bone marrow transplantation before CAR T cell infusion. Just be careful because a very small number. So if the patient is in the first, you will receive the first bone marrow transplantation, probably you will benefit of it. And then also the patient who lose the CAR T cell very quickly, so in the first 6 months. This is also described from other groups. So the patient who lose the CAR T cell in the first 6 months, most of the time they relapse after the infusion. Here the experience also from a Chinese group who had a CD28 CAR T cell, so most of the time this patient and the trial try to get this patient to a second or a third bone marrow transplantation because of the lack of persistence of CD28 CAR T cell. But then you have also an experience from Park and Hall in Sloan Kettering with adult patient who, as you can see, there is really no benefit to consolidate the patient after CAR T cell therapy. So at the moment, I think most of the physicians agree that if you ever you lose your CAR T cell very soon, probably you need a consolidation with bone marrow transplantation. So just a slide on CNS. Here some experience from CHOP and CI. If you can see that the activity against leukemia and CNS it's very good, and was an exclusion criteria in the first trial of the Novartis study, but I think that now everyone is aware that you can have a very good response in this kind of relapses, and also in extramedullary relapse that are not CNS, like testicular relapse or other chloroma, you can have really a wonderful response. Just so until now everything is amazing, but of course you have side effects also in CAR T-cell. As you can see, probably the more important are CRS and in CANs, so the neurotoxicity side effect that are very scary for the family. When you see your child with encephalopathy for 2 or 3 days, it's not easy to deal with. In Seattle, they set up a strategy to try to avoid that. At the beginning, nobody knows if steroid therapy or tocilizumab given too early was not affecting the CAR T-cell expansion. So they set up after the infusion of around 20 patients a strategy where they give very quickly tocilizumab, and if the patient was not responsive to toci, they give very quickly also dexamethasone. This was for fever, but also for low blood pressure or for hypoxia. So they were able to show that if you do this early intervention, they were able to cut off the severe CRS from 30% to 15%. And what was very important was that this strategy was not affecting the expansion of the CAR T cell, not affecting the production of cytokines that are needed for the CAR T cell expansion. So this was a wonderful paper that was published in Blood last year, explaining the physiopathology of the CRS. As you can see, when you have the T cell activation, GM-CSF is released from the T cell, and then you have an activation of myeloid cell. And this is very important because the myeloid cell who produce interleukin 6, interleukin 1, and the other cytokine that are responsible of cytokine release syndrome and neurotoxicity, and also interesting this monocyte activation inhibits the CAR T cell function. So they were able to do a study in vivo, in mice, and what the strategy was to block this GM-CSF production with an antibody, and then what they were able to do is that there was a better functionality of the CAR T cell and a lower toxicity of the CAR T cell. So thank you very much.
You can take any questions. I can have one question.
You can go ahead.
Yeah, you go.
I wanted to ask you, with the dual CAR T, in the responders, is there negativity in both targets, or there's a preference to target one versus the other, like happens with cord blood, for instance?
But I think the CD19 have something special, because we know that the remission it's very good. The CD22, the problem is that you have really this down regulation with lower density. So I don't know if this dual CAR, we have to wait until the study have a longer follow-up, if we are really able to avoid CD19 negative, because we have target CD22. But like in the trial from Seattle, they get problem of the expansion of the CD22 CAR. They have not exactly the same expansion as the CD19. Probably there is something special in CD19.
Thanks, Dr. Francesco, for this great presentation. As you shown here, treatment with CAR T-cell associated with significantly lower CR rate in comparison to the naive CAR T-cell. And also you show that hematopoietic stem cell transplant post CAR T-cell is significantly better. So do you recommend to, instead of retreatment with CAR T-cell, to subject those who failed the first CAR T-cell to transplant?
As I saw before, I don't think, I strongly believe that probably in 5-10 years we will be able to just lift CAR T-cell. Until now we have to do a little bit more research on that. Probably if we have better starting T-cell that will persist like memory T-cells, maybe we'll be able, maybe with the humanized and a dual target we will be able. What now it's, I don't say recommended, but if you lose your CAR T-cell in the first 6 months and you still have MRD who is negative, probably most of the physicians in the field will say to you to go to transplant if you have a donor, if you want to go. But sometimes, you know, it's the second or third relapse after second bone marrow transplantation, the family say to you, you know, no more bone marrow transplantation. So you can have patients who get a lot of, or got a lot of CAR T-cell infusion for this reason. But if it's your first bone marrow transplantation that you will receive after the CAR, probably it's the better solution. And the other point is now a lot of people, it's apheresis the patient as soon as possible. So some teams also apheresis when the patient at a very high risk leukemia, they already apheresis, they froze down the T cells, and then they will use it if it's needed. That as some groups do for also autologous transplant in some solid tumor that are very aggressive.
Thank you. So if no more questions, we will move to the second talk by Dr. Nabil Ahmed. He is the Professor of Pediatric Hematology Oncology and the Professor of Cell Therapy in Texas Children's Hospital, Baylor College of Medicine. He will talk about the engineering of T-cell for immunotherapy of cancer.
Certainly, I'd like to thank Dr. Shadi Father for his kind invitation and the organizers. It's a very nice opportunity. I cannot but say I'm very impressed with the hospital, and I would predict substantial leadership in both the intellect and the cure rates of children in this region, and I think hopefully also in the Middle East. I'm part of a specialized center called Center for Cell and Gene Therapy that spans Baylor College of Medicine, Texas Children's Hospital, treating children, and Houston Methodist Hospital, which treats adults. My interests outside of this have no conflicting relationship pertaining to this work. I would like to speak with you about generally about chimeric antigen receptors. So thanks to Dr. Ceppi, I've had a fantastic background to build on. I would like to analyze with you some preclinical and clinical trial results originating from our group that we've used to better engineer therapeutic T cells, specifically CAR T cells, both in the lab and in clinical trial design. These are namely the tumor heterogeneity and antigen escape, as well as Dr. Ceppi alluded to, the CAR T cell bioavailability after infusion, and some identified problems with immune checkpoint inhibition that mandates us to deliver a product that is protected from this inhibitory mechanism. I would like to add some trivia to the very complete background that I had before. Second to last decade of the last century, two gentlemen worked together, a graduate student and a professor in the Weizmann Institute, and their interest was to study the signaling downstream of the T-cell receptor, the natural T-cell receptor. So to do this, they developed a tool where they joined the zeta chain, which is the main signaling domain of the T-cell receptor, to the variable light and heavy chain, which is the recognition domains of an antibody, creating the molecule that was later called chimeric antigen receptor, short as CAR. Serendipitously, the SOTFA, they conferred on the T cell the distinct specificity of the antibody, the monoclonal antibody they used, and by doing this, they tapped into a very deep evolutional ability of the T cells and their adaptability to killing, calling this CAR T cell that kills differently in a manner that the tumor is not resistant to, that can have distinct specificity, that can be programmed depending on the CAR molecule that you design based on the monoclonal antibody, but it's also a living medication, so it can move and multiply. These were called CAR T cells. One point I would like to allude to that these cells have been freed from the restrictions of the major histocompatibility complex, and in details, this is a tumor cell that has a surface molecule that is intended to be targeted. For a natural T cell or a transgenic TCR T cell, this antigen has to be taken up, it has to be broken down by proteasomes into immunologically noticeable units, and then presented on MHC class 1 on the surface of the cell for the T cell to be able to recognize and kill this target cancer. Unfortunately, cancer cells do a very simple mechanism that either eliminates components of the HLA molecule or the processing machinery. So you can find tumors that are abundant in anti-tumor T cells that are not able to recognize and kill this tumor. In contrast, CAR T cells are able to recognize the antigen in its intact form without any processing, including non-protein antigens, which makes them much more advantageous, especially in solid cancers. About 20 years after the discovery of CARs, we joined the rush into making CAR T cells because of the promising data. We took an antibody fragment from, described first by Winfried Wells in Speyer in Germany, joined it into the data chain, and also included a C28 signaling domain to make a second generation CAR. In extensive testing, we showed distinct specificity against bulk tumors as well as stem cells, and studied this in several models as shown here, and used this as justification to present to the FDA a series of clinical trials. The ones with OS are osteosarcoma that develop into sarcoma trials, and the ones that follow are brain tumor trials, mostly solid tumors, although I'll show you some conceptual work that we've done with Dr. Hisham Azim and LA Children's that I'll present tomorrow. So I'd like to summarize the concepts we derived from these trials, but also the limitations. This was a HER2 targeting trial in glioblastoma, and this trial we infused autologous HER2 CAR T cells in 17 subjects. 7 of these were children, and used the scheme on the stairs I'm showing here to escalate the doses. We didn't see any severe adverse events, and learned a big lesson. This is a scale of 6 weeks. At the end of 6 weeks, we found no particular expansion of the living medication that we gave. This was a big advantage. We hoped that this would expand in the peripheral blood, which did not happen, and also limited persistence. So the kinetics proved to be very disadvantageous. In this trial, we also noticed that patients after infusion within 6 to 12 weeks have an increase in the edema of the tumor that we initially called as progression. Interestingly, when we looked at the swimmer plot, these patients in blue here that were discharged home as being progressive took very long time to die. So now we are revising our criteria for judgment of the tumor progression. At the end of this trial, we had 1 partial response, 7 stable disease with between 3 and 34 months, with 3 patients surviving 34 months with in glioblastoma. This was very interesting because shortly after we published our work, Marcella Maus and her group published a paper in Science Translational Medicine describing a single dose of peripherally infused CAR T cell directed against a target in GBM called EGFRB3. She infused 10 adults and similarly showed no severe adverse events, and some data about positive traffic into the tumors when the tumors were taken out, described some inhibitory molecules as I will show below. So although the trial looked like it is not showing anti-tumor efficacy, we learned quite a bit, and it was very reminiscent of our trials. I think you'll find this curve very reminiscent of the previous curve of very poor expansion and very poor persistence. And the MRIs are very reminiscent of our MRIs of when the tumors getting more edematous between 6 and 12 weeks from the infusion. And the swimmer plot was also very similar to the swimmer plot that we showed, with a lot of patients surviving surprisingly longer after infusion of CAR T cells. So if we tally what lessons and challenges we have, to the left are our achievements, that there are targets described by us, like HER2, alpha-3 receptor alpha-2, as by others like EGFR-3 and alpha-3 receptor alpha-2 in the clinic that seem to be safe to target. GD2 and CSPG4 are being targeted now clinically. We had some evidence of bombing of the CAR T cells with brain tumors. We didn't see toxicity, but when you don't see toxicity, question how efficacious your modality is. So this was questionable. We saw some efficacy with prolongation of survival as well as stability of disease meaningfully. But we also questions how we monitor the tumor, because all tumors looked like they were more edematous. And now we're adopting the IRANO criteria, which excludes the edema from tumor assessment. More importantly, we identified the challenges, and this is our ongoing work. I'll actually start with number 2. I apologize for this mistake, which is the poor CAR T cell expansion and persistence, like Dr. Ceppi alluded to in leukemia, and then follow with tumor heterogeneity as and the antigen escape, and then lastly close with what our work is, and it's the least developed in checkpoint blockade. The first problem is the bioavailability of CAR T cells. This is a curve of the survival of cells in the peripheral blood of patients in the osteosarcoma trial. Independent of the CAR T cell dose, the curves will always going downhill, so you're losing the cells progressively after they're infused. And by about 6 weeks, these cells are completely lost. So we created a lymphocyte space using fludarabine and cyclophosphamide. And interestingly, in the lower curve here, after this flucide conditioning, we had very high levels of the homeostatic cytokine IL-15. And this was associated with about 1,000 to 10,000 fold expansion of the cells as seen here, compared to the previous trial. We called this trial HEROES 2.0 because it had lymphodepletion. Interestingly, this trial was very promising, with one patient with rhabdomyosarcoma going from a disease load shown on the top row to complete remission and recovery of the bone marrow in the bottom row. This trial in total had 10 patients so far that were analyzed. We're still accruing. We've accrued a couple of patients more. Out of 10, 2 were in complete remission for more than 3 years. 4 have stable disease, and 4 had progressive disease. And this is was presented in the ACR and is now in review. What to do with brain tumors? Along with this, we've matured some data working with sick kids in ependymoma and found surprisingly high level of HER2 and ependymoma that is very close to breast cancer. Although it didn't reach statistical significance, HER2 low tumors tended to do much better than HER2 high, and the numbers of patients are fairly high in this brain tumor, and this outcome was associated with independent of where the tumor was, if posterior fossa, port, or supratentorial. When we implanted these in sick kids in Toronto with these tumors and treated them with HER2-specific T cells, we find significant prolongation of the survival of these mice and found an advantage to local regional delivery. So we learned 2 major lessons from this, and that helped us to design the next clinical trials. This data was done in collaboration with Impress now in Nature Medicine. What we learned was the resolution number 1. We presented this for Pediatric Brain Tumor Consortium as a concept that is now in review. It's a phase 1 trial of autologous HER2 CAR T cells for patients with refractory recurrent ependymomas. This is a transplant model, so patients get conditioned using flucide, then they receive their autologous CAR T cells in incremental doses as shown here. This study is not yet open, but to bridge the homing capacity of the CAR T cells, we've now included children on a previously open trial called ICAR, short for intracranial CAR T cells, that is recruiting. We've already infused 2 patients, 2 children with ependymoma, and 3 adults, 2 with breast cancer and 1 with gastric cancer with brain metastasis. This infuses incremental doses as shown here to the bottom right with in an Ommaya reservoir or intraventricularly. We're also allowed to give intrathecally. This is a tissue agnostic trial, but we stratify patients into HER2 amplified like gastric and breast cancer patients and HER2 non-amplified like GBM and other brain tumors. And again, this is given intraneurotomy. The second problem we faced that we learned about from our preclinical work as well as some of other groups' clinical trials is shown here. Mina Hegde joined my lab as a fellow some 9 years ago, did a simple experiment. This is a glioblastoma cell line. She treated this with T cells that are targeting HER2, small doses every day, and showed that HER2 is lost after 15 days with a viable tumor that can actually be tumorigenic, but IL-13 receptor alpha-2 was maintained. So antigen loss at its florid form. We also looked back at the mice that we treated with HER2 CARs and found complete HER2 loss in relapsing tumor, and when we reversed the experiment by giving IL-13 receptor alpha-2 CARs, we found that IL-13 receptor alpha-2 loss. This was interestingly shown a couple of years later by Christine Brown in City of Hope. This is pre-treatment xenografts using IL-13 receptor alpha-2. After treatment, the expression is almost completely lost. But this was also shown by the trial from UPenn targeting EGFR-V3 that was published by Marcella Maus, showing that EGFR-V3 is lost after CAR T cell infusion. So we led this work by pooling different CAR projects, and we call these CAR pools, in which we would pool half doses or quarter doses of each of the antigens we're trying to target, and showed in several publications that this can offset antigen escape. We then reasoned that we can graft a single cell with more than 1 CAR. We went on 2 and then on 3. I'll present this tomorrow on trivalent CAR T cells in leukemia and ALL, and we found that this enhances T cell activation and increases and optimizes the dynamics of T cell killing. Other groups have extrapolated and built on this quite well, showing that you can employ Boolean logics with this, target an antigen or the other antigen, and an antigen and the other antigen, not the other antigen. And then the last one that Dr. Ceppi also showed here, putting a switch here that only activates the T cells when a small molecule is used. This would be very efficacious and allows for targeting an antigen that could be shared with normal body tissues. Came from our lab as well. Zachary Grada published the first bispecific CAR molecule several years ago. This work targeted IL-13 receptor alpha-2 in GBM as well HER2, and we showed by using subcellular imaging that these cells can form immune synapse that can dock to both molecules simultaneously using a proximity ligation assay, and using much smaller doses can cure the tumors irreversibly in mice. So Mina Hegde has published the first usable one and is now working on a clinical trial to infuse this intracranially. The third line that we're working on, and this is led by Mina Hegde, who trained with me in the lab and now is an independent investigator, targeting immune checkpoint inhibition. Data showed prior to my talk substantiated the role of checkpoint inhibition in improving the T cell survival after infusion. We focus on brain tumors and solid tumors. unfortunately, antibodies, the checkpoint blockers, do not make it in meaningful concentrations in the neuroaxis. So let me consider again the mechanisms in T-cell activation and inhibition as a description of what we're doing. If this is the target cell, there is evidence that the T-cell receptor, by activating the T-zeta chain, or some less convincing evidence or thinner evidence in CAR cells, showing that activation triggers downstream phosphorylation of ZAP70 and PKC-theta in this very activation-like axis that employs phosphorylation. Typically, after this, B7 on a tumor engages co-stimulatory molecules in what we call a signal 2. Signal 2 activates the cell in a way that will make it proliferate. At the same time, and because the immune system would like to contain the activation, B7, the same molecule, is able to engage CTLA4, another system, and this converges on SHP2, which is a central inhibitory molecule that seems to inactivate all the phosphorylation events downstream of the target engagement of the CAR, as well as the co-stimulatory molecules. In addition to this, there is another layer that is very famous together with CTLA4. As you know, there are two scientists, one from Japan and one from the US, that received the Nobel Prize this year for blocking of CTLA4 and PD-1 respectively, which underscores the importance of these inhibitory mechanisms. In addition, there is yet another layer of inhibition that comes from MHC class 1, LAG3, also converging on SHP4, which seems to be a very important molecule. So Mina worked with Daniel Langley, who's a clinical fellow in the lab, and thought that we can target this mechanism, target this pathway. So first, because we're interested in glioblastoma, we looked at 15 adult glioblastomas and looked at their expression of the PD-L1. This is the molecule that engages PD-1 and initially activates, but later on deactivates the T cells, and found that about 40% of these 15 patients had constitutive expression of PD-L1, the inhibitory molecule, upon induction of inflammation by putting these cells in interferon gamma. All of them end up expressing PD-L1. Again, we'd like to validate this in the clinic, so let's examine this study from Marcella Mouse again, EGFRB3. They took out 5 of the tumors they treated and found that in addition to FOXP3 cells, which are Treg cells, there is significant overexpression of PD-L1 on the treated tumors, which could explain the inefficacy of the CAR T cell in this scenario. How about the other side of the story, the other side of the immune synapse? We looked at the PD-1 expression in HER2 CAR T cells that we infused, and interestingly found that there is an overexpression of PD-1 upon T cell activation. So the cells become activated and they are very susceptible to the inhibition by the tumor. So both the CAR T cell and the immune inhibitory molecule on the tumor cell are collaborating to fail the CAR T cell efforts. So we reasoned that, or Mina Hagde and Dan Landy reasoned that, what can we do with this? This is the activatory axis, this is the co-stimulatory axis, and this is the initiation of the inhibitory axis. And we decided to focus on PD-1, PD-L1 axis. We thought first that we would amputate the signaling domain from the inhibitory PD-1 molecule, and we did this. And we thought this might make the T cell immune to inhibition by acting as a sink, engaging all the PD-L1 molecules on the tumor cell. But we thought we can take this further, and rather than having an inhibitory signal, we can copy the C28 signaling domain from here and graft it here, and made a molecule that it looks inhibitory on the outside, but is actually activatory on the inside. And we call this checkpoint reversal molecule, CPR, which also stands for cardiopulmonary resuscitation. So we thought that these molecules could salvage the functionality of the T cells upon engagement of the tumor. These are the results. These are the constructs we made. This is the CAR molecule. Has a separation DNA called 2A sequence, and then the CPR molecule. This is the PD-1 molecule extracellular domain that does not have an intracellular domain, so it's a decoy. It is functionalless. we also made on the same transgene molecules that have inhibitory molecules in the inside, and others that split the molecule between the activation between the CAR and the CPR molecule. In this scenario, the CAR would have a CD3 zeta, so this delivers signal 1 activation, and the inhibitory molecule would provide the complementary signal, which could provide a safety switch. We elected to put both on the same transgene because this becomes much easier to translate into patient trials in the future. This data here shows you that we can express both the CAR and the CPR proportionately using this single gene. The SHP2, the molecule I showed you before, it is very central for inhibition, does not get recruited. The green color is the CPR molecule, but there are no SHP2 around the CPR molecule, which means that these cells have reversed functionally the inhibition. This is the result, the survival data from animals treated with these molecules. The winner of these combinations was this one, the HER2 CD3 zeta, as well as a CPR with a 4-1BB signal domain. So Mina Hegde has already written a clinical protocol, and it's being reviewed by our IRB hopefully soon, and the FDA in preparation for a clinical trial. So I hope I've shown you that CAR T cells can demonstrate some promising anti-tumor activity. Again, in solid cancers, it's not as robust as in pre-B-ALL, but it's certainly promising. Efficacy challenges that we're hoping to deal with between our group and other groups is the T-cell bioavailability, the tumor heterogeneity, and the antigen escape. And I'll show you some work, as I mentioned, with Dr. Hisham Azim, LA Children's done together on ALL using trivalent CAR T cells, the environment inhibition, and lastly, I hope I've shown you that we can perform rational cell engineering by continuously monitoring the clinical trials and getting feedback from these results to engineer and design meaningful clinical trials. First and foremost, I would like to acknowledge the patients because they are really volunteering independent of the outcome, and this is extremely courageous and selfless. Mina Hegde was my fellow, but now has been my partner in the lab for the last more than 5 years. This is the rest of our team, and the funding agency is mostly a moonshot from the NIH, as well as a Dream Team group grant with Sam Baldricks and Standard Cancer. I thank you, and I can take questions.
Thank you, Professor Nabil, for the excellent lecture. Actually, I have two questions. The first one for the trials on the recurrent ependymoma, refractory ependymoma, as well as for the glioblastoma. I know the numbers are pretty small, but did you try to separate with the residual volume of the tumor? Do you have applied the treatment for all patients with no residual disease or those with residual or without residual disease? And my second question about...
Let me answer this
okay.
Because I have a very bad short-term memory. We did not do this, but we plan to do this in the subsequent clinical trial. This has been proposed to the Pediatric Consortium. It's much easier with sarcomas, so we're hoping in the next few trials it would answer this question by taking tumors out after treatment.
And my second question about the blood-brain barrier. How do you overcome the concentration of the CAR T cell after the blood-brain barrier?
So far we are not able to meaningfully reach it. There is evidence from Marcella Mouse that after he infused the cells, she infused 8 patients and took out 5 tumors. And I believe in 4 out of the 5 there were significant CAR T cell involved. From our study, we saw edema with the majority of patients we infused had some edema that accentuated significantly after the infusion. So far, the effective way we're dealing with is by infusing the cells intracranially. Yes,
thank you, Professor Nabil. I have a question and a comment. My first question is about the PD-1 expression in the tumor. Is there any plan to combine the CAR T cells with the anti-PD-L1 or chemotherapy immune checkpoint inhibitor, or following the CAR T cells with the anti-PD-1?
Yes. I'm not professor. I'm still associate. So thank you for the professorship. But it is very important that we saw in, hopefully, I couldn't show the data because of the limited time. We saw significant improvement in the kinetics of T cells when they're combined with more persistence. And I think this would be a very promising innovation, a very promising addition to the protocol. We are now, we've now changed our protocol for sarcomas to include PD-1 inhibitor pembrolizumab as part of the consolidation, so after the CAR T cells, not with the CAR T cells.
Yeah, my second comment about the tumor microenvironment, and how I think this is the main challenge in the solid tumor, in the CAR T cells for the solid tumor. What are the planning, or what are your thoughts about to overcome this heterogeneity in the microenvironment around the tumor cells itself?
You mean the heterogeneity of the antigens?
No, the tumor microenvironment. How can the CAR T cells overcome the tumor microenvironment?
There are several. There is a cellular component to this. So convincingly shown by several groups is this. There is an enrichment for Tregs. The lymphodepletion, especially using fludarabine, tends to deplete these Tregs. So this is one axis that is produced, that is provided by the lymphodepletion. Others in our group have engineered the cells to be more resistant to checkpoint inhibition, like I showed now. Others, our group, Malcolm Brenner and our group at David Spencer, have engineered the T cells to not respond to TGF-beta, which is a very immune inhibitory molecule that is secreted by the tumor, such as GBM and pancreatic cancers, and so on and so forth. Others have created what they call the super CAR or the muscle CAR, which is a CAR T cell that can take, that has more cytokine, particularly IL-12 from Sloan Kettering, and those that can take the inhibitory cytokine to deliver a positive signal, akin to what we've done with the checkpoint blockade. They're different. There is a very nice renaissance of engineering T cells that is going on.
Thank you.
Thanks, Dr. Nabil, for this great presentation. Actually, I have two questions. The first question, you mentioned that one of the challenges or problem facing CAR T cell in solid tumor is the tumor heterogeneity and antigen escape. So, is there any prerequisite like testing the pattern of the specific antigen suppression in tumor sampling before giving the targeted T cells? This is the first question.
Yes, let me answer this. So I agree with you. It seems like we tend to treat primary tumors when they look kinder, and the tumors look different after therapy. So Matt Campbell, who works in our lab, is looking at particularly this. How do tumors look after they are challenged? What is the profile of antigens that should guide the production of a multi-specific CAR T cell? So the answer is yes. I think it's very important to, and this is also a personalized way of treating patients, I think.
The second question, if there is any data about the role of CAR T cells in high-risk neuroblastoma targeting the GD2 molecules, which is highly expressed on the surface of neuroblastoma cells?
A study published from our group by Malcolm Brenner and Martin Puley, who is now in England, has shown that there is some interesting efficacy to GD2 specific CAR T cells in children. Small number of children, but a few responses that were, I think, very promising as well.
Thank you. So if there is no any other question, we will move to the following talk by Dr. Mahmoud Hamed, Associate Professor of Pediatric Hematology, Oncology and Transplant at the National Cancer Institute, and a consultant of Pediatric Oncology and Transplant at Children's Cancer Hospital 57357 Egypt. He will talk about the diagnostic and the treatment strategies of juvenile myelomonocytic leukemia.